Okay, let us start today's class. I will start sharing my screen. Okay, hello everyone. Today we are going to deal with the fifth week of our PMRF-NPTEL session on Introduction to Developmental Biology. So these are PMRF-NPTEL sessions in which we solve the previous year's problems or quizzes of the same course to get a better understanding of what the concepts behind the entire course is, as well as help us to solve different quizzes that we are faced with this year, as well as with the exams and everything else. So I will quickly introduce myself. My name is Anuvab Chatterjee. I'm a PMRF fellow and PhD scholar at IIT Kanpur, and I am taking this course. So how we normally proceed with these sessions is that I will be showing you different quizzes from the previous year's run of the course, and we will try to solve them together. I will ask you to try and guess what the answer is, and then we look at the concept behind the question and then solve them as is. Okay, so this is what we're going to do, and let us start with the first question for today. The question is, Towns and Holfreter dissociated cells of embryos of different organisms and visually examined and tracked motions of these cells when they were mixed to form a heterogeneous mass. During this process, they noticed that the cells sorted themselves in a manner that their relative positions within the mass resembled their positions within the intact embryos. Based on these, which of the following do you think did they propose? The options are selective affinity, differential adhesion hypothesis, differential affinity, and conditional specification. So I will give you a moment to try and figure out what will be the correct answer, and then we will look at the concept behind the question. So you can put what you think is the correct option into the chat. A, B, C, D. Okay, so let us look at it. What exactly was the Towns and Holfenter experiment that they did? So these people were looking at how cells associate themselves after they have been taken out of their original embryonic context. So they were working with xenopus embryos, and a good thing about xenopus embryos is that if you take a tissue from the xenopus embryo and put them into an alkaline solution, then the cells by themselves dissociate and form single cells. So they are no longer form a, like are associated as a tissue. So they took two differently pigmented embryos of xenopus and took different parts of the embryo. So from one they took what is known as the presumptive epidermal cells, so that will grow to form the epidermis, and from the other they took the neural plate cells. So obviously these are of two different origins. They dissociated the cells with alkaline solution and formed a single cell mass, and they pooled all the cells together. Once they pooled the cells together, they saw that something interesting happens. They spontaneously re-aggregated. So they formed a ball of cells, and this ball of cells actually was able to resegregate into the neural cells being inside and the epidermal cells being outside. So the idea here is that these cells somehow were able to maintain what association they had before they were dissociated. So the epidermal cells came back together, the neural cells came back together, and interestingly, they aggregated in a way that the neural cells were inside the epidermal cells, which is a similar situation as it happens in the embryo. So not only did they aggregate within themselves, but they also re-aggregated in a way that the cells which were inside in the embryo were inside, and the cells which were outside in the embryo were outside. So this is one thing that they saw, and then they took it a step forward, and they, from the same organism, took different cell layers from different tissues. So they took the epidermis and mesoderm and saw similar things. So once they reaggregated, they sorted themselves out into the epidermis being in the mesoderm being inside and the epidermis outside, similar to how it happens in the embryo. Similarly, with the mesoderm and endoderm, you have the mesoderm inside covered by the endoderm. Similarly with everything else, they reaggregate and seek out cells which are similar to each other and then sort themselves as if they were in the original embryo. So again, another example: neural plate, mesoderm and epidermis. The neural plate goes the most inside, followed by the mesoderm, and finally the epidermis. Exactly how it happens in the embryo, and they only associate with cells that are similar to each other. So based on this, Townsend Holfrener got the selective affinity hypothesis, which states that cells which are similar to each other will have affinity towards each other, and that is why they aggregate together. So that is why the mesoderm only aggregates with the mesoderm, neural plate only aggregates with the neural plate cells, and epidermis with the epidermis. So this is known as the selective affinity hypothesis that Townsend Holfrener built up depending on these experiments. We will look into why these guys sort themselves like they are present in an actual embryo in further experiments. So Townsend Holfrener experiment, what they found out was selective affinity between the cells. Okay, if this is fine, then we'll move forward. Okay, moving to the next one, we'll look at the reason why these cells actually segregated as they are, as if they are in the original embryo. The answer is cadherins, which we will look into in a bit, but first we will try and answer this question and see if we know a bit about them. So identify the correct statements regarding cadherins. The options are: a culture containing con containing two cells containing identical amounts of two different cadherins, say N cadherin or E cadherin, will randomly sort so that one group of cells is internal to the other. The higher amount of cadherins on the cell surface, the higher the surface tension. Neuronal formation is adversely affected by loss of E cadherin but not N cadherin. Cells containing a higher number of homotypic cadherins, like similar cadherins, will sort to the center compared to a same number of heterotypic cadherins, which is like all the cells express different types of cadherins, but the total number is the same as the cells expressing the same type of cadherin. And this option tells us that the ones having the same type of cadherins will sort to the center, whereas the cells expressing different type of cadherins will aggregate to the outside of the entire ball of cells. And the final one is cadherin proteins are required to form tissue boundaries. I will give you a second to figure this out and try to answer this. Okay, let us look into it. Uh, yes. So first, let us look into what cadherins are. So cadherins are cell adhesion molecules. Their name essentially comes from that, and the name is interpreted as it is calcium because they require calcium for their adhesion. We have an answer. We'll look at it in a bit. So we have calcium and then adhesion molecules, so cadherins. So what are cadherins? These are cell surface molecules that have an intercellular part, transmembrane, and then a big extracellular region. So what happens is that these cadherins have different parts in the extracellular region, and the furthest from the cell surface actually has a adhesion site. This adhesion site can bind to other cadherins on other cells. So as you can see right here in the bottom picture, this part actually associates with the cadherins on the other hand, like a pretty big handshake between the two cells, and that is why what causes the cells to attach to each other. On the intracellular part, they are associated with the actin cytoskeleton. So actin cytoskeleton is essentially what is holding the cell up to essentially holding the shape of the cell, and they are connected to the actin cytoskeleton via molecules known as catenins. And further, the cadherins can be regulated on how much they can adhere to other cadherins or do not cannot adhere to other cadherins, depending on phosphorylation on the catenin binding sites on the intracellular domain. So cadherins are cell surface molecules which help attach to other cells or the extracellular matrix by linking to them by their extracellular domains. So this is what cadherins are, and this is the reason why cells actually can sort themselves in the aggregation that we just saw. So it's the first example of cells actually sorting and actually trying to explain the phenomenon of the cells sorting by themselves, what is by Fortis proof of Malcolm Stigger's differential adhesion hypothesis. So he hypothesized that cells will sort randomly or spontaneously because the surface tension on the cells is different to each other. So a type of cell such as the limb bud here shown in green has a higher surface tension compared to the red cells here, and that is why the limb cells will go inside while the red cells stay outside. So higher the surface tension, the more the tendency to be towards the center of the entire structure. So any cell type with a higher surface tension will always go to the center, while the other, which is of a lower surface tension, will stay on the outside. So the differential adhesion hypothesis tells us just this, that these cells actually segregate due to higher or lower surface tension. So cells sort to form aggregates within the smallest interfacial free energy. So essentially allowing the most highest surface tension cells to go inward while the lowest ones stay outward. And why does this happen? This happens because of different amounts of cadherins present on their surface. So this is shown right here. So this is one of the experiments that was done. So these guys, what they did is that they took different amounts of cadherins and expressed it in different amounts in different cells. And what they saw is that as the surface cadherins increase, the number of cadherins on the surface of the cell, the surface tension also increased. As you can see, pretty straight line, more the surface cadherins, more the surface tension, and as we saw just now, that more the surface tension is actually the driving force to actually cause the cells to sort themselves. So cadherins and the number of cadherins helps in determining the surface tension, and that surface tension causes the cells to sort themselves. So going back, identify the correct statement regarding the cadherins. So we go one by one. So a culture containing two cells containing identical amount of two different cadherins will sort randomly, with one group of cells being interior to the other. This is true because cadherins within themselves have a particular sort of interaction which causes them to attach strongly to each other, increasing surface tension, and that is why you will have one group of cells going interior, one staying outside. The higher amount of cadherins on the cell surface, the higher the surface tension. This is also true, we just saw that with the graph. Cells containing the same type of cadherins will sort to the center as compared to the cells containing different types of cadherins, because again The same type of cadherins have a stronger interaction between them, increasing the surface tension, and those cells will go towards the center, while those cells having different types of cadherins will have less strong interaction, so lesser surface tension, they will stay on the outside. And cadherin proteins are required to form tissue boundaries, which we saw from all of these examples. The cells sort themselves back into their original tissues, and there will be examples we will see with the neural tube as well, in which you will recognize that specific tissues express specific type of cadherins so that they can attach to each other and form the entire tissue. And that is what the tissue boundary, that is how the tissue boundaries are formed. So we have so many correct options. So Desi Hari actually told us that it is B. Yes, you were correct. B is the correct option, but there were also more. So any questions from this? Okay. Question along the same lines. Based on the figure, choose the correct order of surface tension. So what do you think is the correct, like which cells have a higher surface tension? So you see the same image here, in which green and red, when they associate, green goes in, red is outside. Red and yellow, red goes in, yellow is outside. Yellow and blue, yellow goes in, blue is outside. And blue and orange, blue goes in, orange is outside. So what do you think is the order of the surface tension? So you have four options: green, red, orange, blue, yellow. You choose which one you think is the correct option. I will again give you a moment to figure this out. Okay, so Hari says it is D, green, red, yellow, orange, and blue. Okay, there is an extra more than sign for some reason. Okay, so this is what we just saw: Malcolm Stinger's differential adhesion hypothesis. The higher the surface tension, the more the tendency that the cells will go to the interior of the entire mass. And so if we look at this, we see that when green and red associate, then green goes in, red stays outside, so green is greater than red. Red and yellow, same thing, red is greater than yellow, yellow blue, and blue orange. Essentially just, this is just the order. So green, red, yellow, blue, and orange. Green, red, yellow, blue, and orange. Yeah. So the correct option is number B. The, yeah, it should be number B. I don't have arrow here. No. So the correct option is option B. Green, red, yellow, blue, and orange. Any questions from this? So the one that goes to the interior has higher surface tension. So because, and then we just saw this diagram as well. You can see that the surface tension is just given here. So 20, 12, 8, 4, and 6. A 1.6. So this is just the order. Okay, moving forward. So again, yeah. So again, a question on the cadherins. Identify the correct statements regarding cell-cell communication. The options are: cadherins are linked to the actin cytoskeleton, M cadherin is linked to microtubules, catenins associate with cadherins on the extracellular side, cadherins are phosphorylated at their catenin binding sites. This again is a more than one correct type of a question. So there are, you can choose more than one if you think they are correct. I will give you a minute to figure this one out. Okay, so we have answer. Desi Hari again says it is A and C cadherins are linked to the actin cytoskeleton, and catenins bind to cadherins on the extracellular side. So let us look at it. So this is the image we just saw. So cadherins are on the cell surface. They have an extracellular side which binds to other cadherins, and an intracellular site. They are regulated with phosphorylation on the catenin binding sites, and they are associated with actin. But the catenins are the mediator essentially between the cadherins and the actin cytoskeleton, and they are present on the intracellular site. So this is how entirely the structure looks like. Cadherin on the cell surface, followed by catenins, followed by the actin cytoskeleton. Phosphorylation occurs at the catenin-cadherin junctions, and cadherins use their extracellular part to bind to other cadherins or integrins or other adhesion molecules. So let us look at it. The answer is cadherins are linked to the actin cytoskeleton, and cadherins are phosphorylated at their catenin binding site. The option C is not correct because it says that catenins associate with cadherins on the extracellular side, but they actually do it on the intracellular side. They are the mediator between the cadherins and the actin cytoskeleton. They are on the intracellular part. The extracellular part actually only associates with other cadherins on the outside. So option C is not a correct option. It's A and D. Is it fine? Okay. Okay. Moving on. Which of the following types of cells are found by cells of the ventral furrow during Drosophila gastrulation? The options are endoderm, mesoderm, ectoderm, and germ cells. So, if you have a clue about how which uh, cell layer forms which part of a particular organism, then you will have a pretty easy time answering this one. So endoderm, mesoderm, ectoderm, and then germ cells. Which of these form the ventral furrow? So the ventral furrow is essentially is the part of your the front part for us, the belly side. So which of these is going to be the correct option? Okay, DC Hari says that it is option B. Let us look at it. So this is what Drosophila embryo looks like, and this is the ventral furrow, which is the belly side for us. And here cells actually invaginate and move inwards. So this is actually part of the mesoderm. You're correct. So as we remember, obviously that it is ectoderm, mesoderm, endoderm, the three layers. The ectoderm forms the surface skin layer, the epidermis, as well as the neural, all the neural part of the central nervous system, and the mesoderm forms all the internal organs and all the tubes, the entire uh, esophagus, the entire food pipe is formed by the endoderm. So anything which resembles an organ actually always comes from the mesoderm. Okay, moving on. Now we go into a very interesting concept known as competence and induction, and we just introduce it with the following question, which is organize the following in the correct sequence of events. The options are competence, development, and induction. So what order does the entire process go? So you can choose 1, 2, 3, the the order in which it occurs. The options are given right here, 3, 1, 2, and then combinations of these. So you have to choose one of these. So I will give you a second to try and answer this. So we get the option B, which is 1, 3 and 2, which is competence, induction and development. Okay, uh, let us look at it. So we'll go through a bit about what exactly is competence and induction. So all the cell types that we have are actually, when they develop into something, undergo different stages. So particular cell type or a tissue type needs to be induced to form a tissue. So it receives some kind of a signal from the neighboring tissues so that it can form a particular structure. But the interesting thing is that not every tissue will respond to this inducing signal. The tissue needs to be competent enough to uh, receive the induction signal and then form whatever the tissue it needs to form. So the best example to deal with this that we even have in the book is the optical cup formation. So okay, this particular schematic shows a cross section. I am not visible here. I'll place it here. Okay. This particular schematic shows a cross section of an early Xenopus embryo, and this is the head region or the presumptive head region. And we are talking about this region in particular. So this part you can see right here. This is known as the optic vesicle. Optic vesicle induces or gives out signals to the overlying cells here, which are actually the surface ectoderm, to form something known as the lens vesicle. So this essentially will form the lens of the developing eye, and this is formed from the surface ectoderm. Now people asked, the obviously these two tissues are under like over each other, and it why does the optic vesicle cause the lens to only form here? This is the question that they asked. So they said, okay, what the best way to go about is that we dissect the optic vesicle out and then place it somewhere along the embryo which is not close to this region which normally forms the lens. When they took the optic vesicle out, put it somewhere else, they saw that optic vesicle here could not induce the surface ectoderm here to form the lens. So this led them to believe that if the optic vesicle, so this was experiment 1, and the other thing that helped along their ideas is that when they take the optic vesicle out, they form no lens. So obviously this gives them the idea that okay, the optic vesicle is sending out some signals to the surface ectoderm, which is here, to form the lens, because without the optic vesicle then we have no lens. So obviously optic vesicle is a inducer. It is sending out signals and inducing the surface ectoderm to form the lens. But when I take the optic vesicle and place it somewhere else, it cannot do the same. It cannot cause the surface ectoderm to form the lens. Here the next idea came. So optic vesicle is an inducer, so it induces the surface ectoderm to form the lens. But all over the embryo, the surface ectoderm is not competent to form the lens. So even though the optic vesicle is sending out signals, this particular surface ectoderm here, all over here away from the head, is not actually able to form the lens. So it is not competent enough to receive the signal and undergo lens formation. So these two things go hand in hand. One tissue induces the other to form something, and the other tissue needs to be competent enough to receive the signal and form whatever structure it needs to form. So induction, inducer or responder. So responder is going to be the surface ectoderm and inducer is going to be our optic vesicle, and competence is going to be the ability of the surface ectoderm to receive the signal from the inducer and form the lens. And they also confirmed this with a final experiment in which they took a tissue which was not a optic vesicle. They took a tissue from elsewhere in the embryo and they all transplanted it near to this lens forming surface ectoderm, and they saw that also the lens did not form. So obviously in this case there was no inducer present again. So it was as if the optic vesicle is not there and no other tissue can replace its function. So any other tissue being here, again it is the situation where the uh, inducer is absent. So even though the competent cell layer is here, no one there to send out the signals and induce them to form the lens. So inducer, the tissue or the cells which sends out the signal and causes uh, neighboring tissues to change their structure and undergo morphogenesis. And competence is the ability of this cell layer which is undergoing morphogenesis to actually receive the signal and do so. So cells surface ectoderm competent, optic vesicle is induced. It is an inducer. Is this fine? If you have any confusion, please let me know, because this is like a really important part which we will have to go back to again and again later on. Okay, I believe then this is fine. So going back to the question, what is the order of the sequence of event, competence, development, and induction? The order is competence comes first, because if the overlying surface ectoderm is not competent, ah, yeah, I can explain again. Okay, we go back. So competence and induction. So it is. I will give you an example. So it is like someone throwing a ball and you catching it. Essentially, if we imagine induction and competence in that way, then whoever is throwing the ball is the inducer. He throws a ball towards you and that allows you to catch it. But you have to be competent enough to make a very good catch. Otherwise, there is no point in throwing the ball. They can throw the ball at someone else who is not at all very good at sports, and you throw 100 balls at them, they cannot catch anything. So that is the essentially the interaction between an inducer and inducer cell and a competent cell. Inducer cell sends out a signal, competent cells receive it and do something. If the inducer is not there, doesn't matter, there is no one to throw the ball, doesn't matter if you can catch it. So without the inducer, no competence or competent cell does not matter. But without the competent cells being there is no point in throwing 100 balls, because they will not be able to do anything with it. So even if the inducer is there and the competent cell type is not there, or the cell type is not competent, then it will not actually undergo morphogenesis. So in the example, our optic vesicle, this layer here is the inducer, throws out the signal or throws out the ball, and it is received by the surface ectoderm here, which receives the ball or the signal and forms the lens. So how does the entire thing go? The first thing is that they remove the optic vesicle. So optic vesicle is gone. So even though the catcher is there is no one to throw. So again, there is no changes here, because there is no one to induce the change here. Even though they would have caught the ball if someone threw it at them, no one to throw the ball. And then if they transplant a different tissue, this guy cannot throw the ball at all. So this, even though again there is a catcher present who will catch it, if you throw it correctly, this person cannot throw the ball at all. So again, even though there is a competent cell layer here, if there is no proper induction or a cell type that induces the change, there is again no lens formation. And lens formation is this invagination of the layer, which is the initiation of the lens. It will later on form the lens of the cell, of the organism. So if there is no thrower or no one to throw the ball, doesn't matter if a competent catcher is there, no change occurs. If there is a person who cannot throw the ball at all, again doesn't matter if a competent cell layer is there, they will not be able to show any changes. And final example is that if there is an inducer, a person to throw the ball, but the other side cannot catch it. So this is the trunk ectoderm which actually cannot undergo these changes because it is not competent. It will not have the genetic makeup or the signals to actually form the lens. So even though the thrower is there, which can throw the ball very well, this trunk ectoderm cannot actually catch it. It is not competent. And when I talk about throwing the ball and catching it, actually I'm making an analogy to cell-cell signaling, which we will see at the end of the entire thing. Cells communicate by throwing out molecules towards each other. So one has to throw the molecule and other has to be able to catch it. If this guy is not able to catch it, it doesn't matter how many times I throw. There will be no changes here, which is what we see here. Even though there is a tissue inducer, the cell layer neighboring it is not competent to form the structure. So it does not have the ability to catch the signals and undergo morphogenesis. And that is why if the inducer is here near the trunk ectoderm, there is no lens formation as we see right here. So this is the entire concept of inducer and competence. One tissue layer sends out a signal that they are the inducer, the other tissue layer catches it and undergoes morphogenesis. The ability to be able to catch the signal and undergo morphogenesis is known as competence. So you have to have both at the same time for morphogenesis to actually occur in such a structure. Is this fine? Okay. So if this is fine, then this question should be pretty easy. Organize the following in the correct sequence of events: competence, induction, and development. So first you need to have a competent cell layer, so someone to catch the ball. It doesn't matter if it is not, they are not competent. Doesn't matter if you induce a lot, they won't change. The first thing you need is competence. Second is you need someone to induce or induction, someone to throw the ball after you know that they are going to catch it. And finally, together both of them cause development to occur, as we just saw: competence, induction, development. If this is fine, then we will move forward. Let me know if this is fine. Okay, moving forward, match the appropriate outcome of each genotype. So this is exactly the same example that we saw with the optic vesicle and the surface ectoderm. The options are PAC6 is present in the optic vesicle or PAC6 is present in the surface ectoderm, finally causing the lens to form. And this is a match the following kind of a thing. So if PAC6 is present in the optic vesicle and not in the surface ectoderm, what happens, and vice versa. So, so that we are able to solve this, I will give you the hint. PAC6 is a molecule which causes competency. It causes this particular cell in this particular scenario to be competent to receive the signal. So, based on that, do you think I will ask you to try and figure out this match the following? So, for example, should it be PAC6 present, PAC6 present in both of them, and lens formation occurring? So, if PAC6, the only thing we are interested in is that it causes competence. So I will let you try and figure it out. Give you a minute for this because there are a lot of options. Okay, we have option A, which is PAX6 present, PAX6 absent in the surface ectoderm, and lens formation occurring. Okay, let us look at it in detail so we are able to understand what is going on. So, as I said, PAX6 is important for for optic lens formation, and it is a competence factor. So, it causes a particular cell layer to be competent. So, we know that we just saw that out of these two guys, the optic vesicle and the surface ectoderm, the surface ectoderm is the competent cell layer, whereas the optic vesicle is the inducer cell layer. So a competence factor needs to be present in the surface ectoderm to be able to receive the signal from the inducer. So if PAX6 is a competence factor, then I only care if it is present in the surface ectoderm. So let us look at it. This is a example in which they did an experiment in which they took two different cell layers from two different genotypes and they put them together. So there is a layer at the bottom which is the surface ectoderm, which is the optic vesicle, and the layer at the top which is the surface ectoderm. So when they took wild types for both, which is normal things for both, we have lens formation. The surface ectoderm was competent enough to receive the signal from optic vesicle and form the lens. When the optic vesicle does not have a PAX6, PAX6 minus, PAX6 minus means it is a mutant for PAX6, and but the surface ectoderm is wild type, that is, it already has PAX6 in it, you still have lens formation because it is a competence factor. I only care if it is in the surface ectoderm and working. If it is there and working, then it is competent and will receive the signal from the optic vesicle. But if you remove PAX6 from the surface ectoderm, then it has lost its competence to form the lens. And doesn't matter if the optic vesicle is wild type, optic vesicle is throwing out the signals, but surface ectoderm is unable to catch it because there is no PAX6. And thus you have no lens formation. And finally, if you have no PAX6 in both of them, again you have no lens formation, but it is majorly because the surface ectoderm does not have PAX6. It is not competent to catch the signals from the inducing optic vesicle, and that is why you do not have any optic lens forming here. So essentially you can imagine PAX6 being the ability of the surface ectoderm to catch the signals from the optic vesicles. Without PAX6, it is not able to catch the signals, or even if it is able to catch the signals, it is not able to act on the signals. So the moment you take PAX6 out, you have no lens formation, doesn't matter what else happens. Is this fine? Okay. So let us look at the options now. The first option, 1, 2 and 1. PAX6 is present in the optic vesicle. So essentially I don't care about this optic vesicle part, if it has PAX6 or not, because this is the ability to catch the signals. So I don't care, because optic vesicle, the role is to throw out the signals. So I don't care about this at all. Only thing I care about is if the PAX6 is present in the surface ectoderm. So the ability to catch is what the surface ectoderm does. So if PAX6 is present or not, and then I can just predict it forming the lens from this. So I ignore the first column, so it is 2 and 1. PAX6 absent, lens formation cannot happen. PAX6 needs to be there for it to catch the signals and form the lens. So not true. 1 and 1, PAX6 presence, lens formation, perfect. 2 and 2, absent, no lens formation, perfect. 1 and 1 again, PAX6 present, lens formation, perfect. 1 and 2, PAX6 present, no lens formation. This will not be the case because PAX6 renders competence, and if it is there and there is an optic vesicle there, then there will be lens formation. So essentially the B, C and D are the correct option. Is this okay? I will give you a moment to digest this. all this, yes, in this particular scenario, all of this depends on the surface ectoderm, because we are talking about a competence factor such as PAC6. So PAC6 is the ability of the surface ectoderm to catch the signals and act on it. So that is why we care about the surface ectoderm so much in this example. If I gave you a different example in which we were talking about an inducer factor, say the ability of the optic vesicle to throw the signals out, then we will be really interested in the optic vesicle only and maybe not the surface ectoderm. But here, competence factor, the cell layer which is competent, is the surface ectoderm. That is why we care about this so much. Yeah. If you have like deeper doubts, then you can just unmute yourself and ask. It's not a problem. Is this fine? Okay. Moving forward. Induction of cell fate, what we have been talking about for the last 10 minutes. What are all of these? What can you state the induction of cell fate is? Is it sequential? Is it reciprocal? Is it synergistic? Or is it mutually exclusive? So this is also a more than one option type question. So induction of a cell fate is sequential, reciprocal, synergistic, or mutually exclusive. What do you think is the correct option, or are the correct options? Whether there is a more than one correct option type of a question? It's B and D, reciprocal and synergistic. Okay. Yes. Let us just look at it. So we have been talking a lot about the optic vesicle and lens formation, but interestingly, the inducing cell layer, as development proceeds further, does not always remain the inducing cell layer. It can be induced by someone else. So, you throw the ball to someone and they throw it back. That can happen. So, this is a pretty complicated chart. We will just look at this. So, we have the optic vesicle. It sends out a signal and induces the prospective lens ectoderm, the surface ectoderm we have been talking about, to form the lens placode. But the lens placode, once it is formed, actually induces the optic vesicle to form the optic cup. Once the optic cup is formed, the lens placode develops to form the lens vesicle, like it is developing, and it now again sends out a signal and induces the optic cup to form the neural part of the retina, and whatever does not is not induced forms the pigmented part of the retina. And finally, the neural retina sends out signals and induces the lens vesicle to form the lens. So essentially, uh, instead of one cell layer always inducing the other, it is always a give and take of a scenario. But it occurs in a sequential manner. It couldn't be that the lens placode formed first and induced the optic vesicle. It always goes in a sequential manner. So optic vesicle, it throws the ball to the lens ectoderm. lens placode throws it back. Optic vesicle, uh, then lens vesicle now throws it towards the neural optic cup to form the neural retina, which throws it back to form the lens. So it is a sequential manner in which all of this development is occurring, and the position of the inducer and the competent cell layer can change, so it can reciprocate. So entire cascade of induction is reciprocal and sequential, both of them at the same time. So if we go to the question, induction of cell fate can be sequential, happens one at a time, like, or in a particular order which is already set, and reciprocal because both of them are all, one person is not always the catcher and the other person is always the thrower. The inducer and competent cell layer can change, the other can induce changes in the other, the first one, once development proceeds a bit further. As for mutually exclusive, it is not mutually exclusive. As we have seen from the example, you take the competent cell layer out or the inducer cell layer out, there is no development. Synergistic, uh, hmm, well, I can be, I can partly agree to that, that you can call it synergistic, but actually the reason being is that sequential and reciprocal is kind of uh, type, or like a way to say it. Like, you can argue that it is synergistic on some of the explanation of the term, but normally people only say sequential and reciprocal. This is more like people have already come before and established what we are going to call it, so sequential and reciprocal it is. Is that fine? Okay, perfect. Yes, this is another example of induction and competence. Which of the following is depicted in the figure? So I will tell you what the example experiment is, so that we are on the same page. So what they were doing is that they, the same thing about induction and competence, but they were looking at different cell layers at this time. So what they did is that they took mesenchymal tissue, which is the underlying tissue, not the skin layer, but the underlying layer on the surface of chicken. And what they did is that they placed the skin layer or the epidermal epithelium on top of that, and they saw what happened. So they joined the wing mesenchyme to the epithelia, and they saw that a wing feather developed from the epithelia. Nice. But when they took mesenchyme from the thigh and then placed the epithelia on top of it, the same epithelia now formed a thigh feather. And finally, when they took the mesenchyme from the foot of a bird and put the same epithelia on top of it, it formed scales and claws. So this is a very interesting thing that they observed, and we are asked to describe which of the following is does describe this phenomenon, which is called regional specificity, temporal specificity, genetic specificity, or epithelia to mesenchymal transition. So I will give you a second to try and figure this out. Okay, the options are A and D is being chosen. So this is a single option type of correct question. You can choose only one. But okay, so we'll look at it. So this is an example of regional specificity. What does regional specificity mean? So as we saw with the optic cup and the surface ectoderm, similar scenario occurs here. The mesenchyme is the inducer and the epithelia is the competent cell layer. So inducer sends out a signal, competent cell layer catches it. But interestingly, Depending on where the mesenchyme is from, it sends out different signals. The wing mesenchyme sends out the signal to form the wing feather. The thigh mesenchyme sends out the signal to form the thigh feather. The foot mesenchyme sends out the signal to form scales and claws. So this is an example of regional specificity. Depending where the epithelia is placed, it is competent to form anything. But which region is inducing it will determine what final developed structure will form. So this is an example of regional specificity. Can we say the specific location of the mesenchyme? Yes, where the mesenchyme is coming from is important. So if we go back to our original this experiment, so you can imagine the optic vesicle, instead of there being an optic vesicle, we are now transplanting optic vesicles from different parts of the body. So different mesenchymes being here, and it is inducing different changes in the surface ectoderm. This is what happens. So here we say that different mesenchymes from different parts of the body actually give out different signals, which causes the competent epithelia to form what the underlying mesenchyme wants it to form. So it depends especially on the location of the mesenchyme. You're correct. Is it fine? Okay. So move on to the next question. This is a big question. So C. elegans vulvar formation occurs during larval stage from 6 vulval precursor cells, VPC cells. The anchor cell connects the overlying gonads to the VPCs. The 6 VPCs form an equivalence group. Each member of this group is competent to be induced by the anchor cell and can assume one of three fates depending on proximity to the anchor cell. The cells directly beneath the anchor cell divides to form the central or the primary vulval cell. So which signaling enables this fate? The options are paracrine signaling, autocrine signaling, juxtacrine signaling, or endocrine signaling. So we have looked at vulval development in the last week's lectures, but here the only thing we care about is that an anchor cell which is a bit further away from all the 6 VPCs, what kind of signal will it throw out for the VPCs to change their fate? Will it be a paracrine signal, autocrine signal, juxtacrine signal, or endocrine signal? So you can put in whatever you think is the correct option into the chat, and this is also a one correct kind of a situation. So any clue about this one? Okay, so let us just look at it. So okay, this here says it is A, paracrine signaling. Okay, good. So let us look at what different types of signalings are. So cells communicate with each other by actually interacting via molecules. They send out some particular molecules so that they as a way of signaling to the other person. So that is why it is called cell-cell signaling or cell-cell communication. So cell send out a molecule which the other can receive. So there are majorly four types of signaling molecules, signaling types. The first is paracrine, where the one cell sends out uh, signals to a cell a bit further away. So if the signal has to travel, it is a secreted molecule. It is known as paracrine signaling. Goes a bit further away. Then there is juxtacrine signaling. Juxtacrine is just when it is the cells touching each other. They have cell surface bound signaling molecules and they touch each other to actually tell something to the other cell. So they signal by touching each other. Like they have to have interacting junctions, and this is known as juxtacrine signaling. Then we have autocrine signaling. Autocrine signaling is when the cell wants to tell itself something. So it sends out signaling molecules that attach to receptors on the same cell, and then it acts upon the signal sent by itself. So that is known as autocrine signaling. And finally is known as endocrine signaling. This the great example we have is called hormones. We have hormones that are secreted from one part of the body that travel all the way to the other part of the body, so hugely long range signaling. So this is known as endocrine signaling. So we are mostly interested in the 1, 2 and 4, paracrine, juxtacrine, autocrine. And paracrine is the one that we focus the most on because this is the most interesting one, which where cells can signal to its neighboring cells via signaling molecules which are secreted. So going back to our question that we were asked, so this is a schematic showing the anchor cell and the, so this is our C elegans, this is our gonads, this huge pink thing, and the black dot is our anchor cell. The anchor cell signals to the VPCs, and these are the VPCs. A blown up image, this is the gonad again, this is the anchor cell, and these are the VPCs. So it has some to somehow signal to the VPC closest to it, which is the P6P VPC. So what does it do? It sends out a signal which is a paracrine signal. So these cells are not touching each other, but these send out a paracrine signal to the anchor cell sends out a paracrine signal known as the protein lin3, which goes and then binds to the P6P, and the closest, since P6P is the closest one, it starts acting first and then forms the primary fate. Then via juxtacrine signaling between these two, the P6P and the P7P, and the P6P and the P5P, it causes them to form the secondary fate. And the cells which are the furthest, which have received no signal from the anchor cell, form the tertiary fate. So anchor cell sends out a paracrine signal. The cell which receives the most causes the other ones to form secondary cell fates while it becomes the primary, and the cells which are on the further apart, which do not receive the paracrine signal, form the tertiary fate. This is again shows another point of the paracrine signaling, that it can the signaling can affect neighboring molecules, and the distance is a matter. So some paracrine signaling signals can reach very far away, but some can only reach, say, 2 or 3 cells apart, and whereas there will be cells which do not receive it directly. So this is how the entire thing occurs. Anchor cell sends out a paracrine signal. So going back, the cell beneath divides and forms to the central vulval cell, which signaling enables this fate. This is paracrine signaling. Okay, any questions from this? Okay, if that is it, then I had, this is all the questions that I had for you today. Any questions from the entire week's lectures? Anything that you have an idea, did not understand or want to get a better understanding of, or any questions from that? Then we can deal with that as well. None. So do you have a question or? Okay. If you do not have any questions, then I would say thank you for joining today. And this recorded lecture will be available on YouTube, and you can go back and watch it if you want. and I will meet you next week. Thank you.